My name is Anson Rosenthal, and today I'll be talking about my research project, Preventing a Zebra Mussel Invasion in the Susquehanna River using a mathematical model. A little bit of background: the zebra mussel is an invasive species of freshwater mussel. It's thought to have been introduced in the United States and the Great Lakes in the late 1980s via the ballast water that cargo ships had to carry over, coming from the bodies of water from Eastern Europe and Asia. So we have a little time series starting in 1988. They're thought to be introduced right around this Great Lakes area. We have a few recorded sightings, and then in 2008 we have many more recorded sightings throughout the bodies of water in the eastern United States, radiating outward from the Great Lakes. As you can see, they have a pretty wide tolerance in terms of water conditions, having colonized all the way down to the mouth or the Mississippi River Delta. So pretty adaptable in that way. They have two vectors of spread, both stemming from the release of what are called veligers. They're the planktonic larvae. There's a long stream dynamic in which these veligers are just swept downstream in the current. And there's also the human vector, in which say an RV or a boat is trailered from a colonized body of water to another body of water that was not previously susceptible, thereby exposing that body of water to an invasion. And once the zebra mussel population has been introduced into a body of water, they can cause two serious problems. One, from an ecological perspective, you have the massive depletion of lower trophic level organisms like phytoplankton, which can destabilize the ecosystem from the foundations on up. And in the Hudson River, it took the zebra mussels only 2 years to reduce the phytoplankton concentration by 80%. So they can outcompete the native organisms, which is a concern for organizations looking to conserve the natural biodiversity. And you also have the economical perspective. They encrust or biofoul municipal water systems and the intake valves and systems of power plants and industrial facilities, which is also a major problem, or would be a major problem in the Susquehanna River. So the objective, I have a two-part objective. The first part is to look at how an invasion might proceed if we do not take any preventative steps. And the second part is, if it is severe enough, potentially, what can we do, or how effective would a preventive strategy be? And the one I propose here is one consisting of low-level continuous chlorination of the water. It sounds scary, chlorinating the water, but it's only it would be confined to small stretches over a small period of time. So it's my hope that it would minimize the collateral damage. So the model approach that I'm taking here is suggested by researchers looking at zebra mussel invasion in the Arno-Sieve River system in Italy. There are a number of differences between my approach and their approach in terms of the details, but I can get to that later. And it can be described as both spatially explicit and semi-discrete. It's spatially explicit in that I can represent the Susquehanna River as a virtual one-dimensional lattice of nodes, each of which correspond to locations on the real Susquehanna River in terms of the water conditions and current speed, which puts the invasion in a more realistic context, so I can project how these mussels would spread in the real Susquehanna River over time. And it's semi-discrete in that there are two timescales running. You have the long-term timescale of years, and that's how I can show how the population as a whole will spread from an introduction point. And then you have the timescale of days, along which the veliger transport is modeled, because the veliger dispersion period is generally between 7 and 28 days. So you also have to take into account how they're going to die, settle, and be produced and travel during that period as they're traveling along the river. So the first part of the model is the adult mussel population growth model. It is a system of difference equations, and here you have 4 equations describing the year-to-year growth of each of 4 age classes of the adult mussel population at a particular location, denoted by X. And this is dependent on these survival rates denoted by sigma, that are biologically unique to each of these age classes. So that's really how I can parameterize this particular model to the environmental conditions that I'm working with in the Susquehanna. So how I did that, I don't really show the particulars here, but this is the end result. I obtained data describing the zebra mussel population density at more or less one location, a very small stretch of the Hudson River, which is geographically similar in terms of the basic water conditions to the Susquehanna River. I found data describing the zero muscle population density growth over 18 years, and I implemented these difference equations on the previous slide in a dynamic spreadsheet in Excel, and used Excel solver to change those biological parameters and match up the model predictions to obtain the closest fit of those predictions to the data. And as you can see, I project the data here in black, and also have the model with that optimized fit in red. And it's very close. You get the same periodic dynamics, which are produced by the cannibalism dynamic mentioned here. No, well, cannibalism, because once the adult mussels reach a critical density, they filter out their own velgers, so there are not enough velgers settling in that year to sustain that density, and that's why you get this periodic rise and fall in population. So once I had that spatially discrete aspect in terms of the adult mussel population growth at distinct locations, I had to integrate that with the current velocity data that I have obtained from the GIS. So I use the USGS enhanced river reach file, which describes the current velocity, along stream distance, and a number of other water factors from waterways all over the United States. And I designated a particular stretch of the Susquehanna River. I chose the one extending down to the Chesapeake from the northernmost reach, because there was a small concentration of velgers spotted in this region right up here in early 2009. So I wanted to see how that might progress, if they're to be seen there again, or if they actually settled and there just haven't been any reported sightings of adults. So I extracted both along stream distances along each of these nodes that were arbitrarily designated by the USGS, along with mean current velocity along the segments. But there was one problem. These are of uneven segment length. So what I had to do is take these uneven segments and interpolate them. I use a linear interpolation, because once you get to higher order interpolations like quadratic or cubic, you end up with these fluctuations, which I didn't find to be physically justifiable. So I just used a linear interpolation. And from there I derived a continuous system that could be then arbitrarily partitioned for however fine a spatial resolution I needed. This became necessary because with the model structure that I was using, it assumed that there was no velger settling in between these specific discrete nodes that I designated. So I needed to make the spatial resolution small enough so I could I could minimize that velger loss as a result just of the structure of the model. And here's the actual velger transport model. It's a partial differential equation with 4 components. You have, the first is the along current spread, which is advection, and this is just the sweeping of the velgers downstream at the speed of the current. You have the diffusive action, just again simple diffusion model. We have the mortality component, where you have mu denoting the natural mortality rate of these velgers over time, and then gamma denoting the cannibalization rate, which is dependent on the concentration of adult mussels at the location. And then you have the external input term, and that's how I created the particular introduction scenario that I did. I used a point spread model in which I introduced a very high concentration of velgers at the first node at the first instant in time, after which they were just spread throughout the system with no other external input. That would simulate an accidental dumping of velgers into the system. Say if somebody's trailing a boat from the Hudson and they dump it into this northern reach. But there was one problem. Since it's a partial differential equation, it's very computationally inefficient to evaluate over such a large computational domain. I had a 585 kilometer stretch of the Susquehanna River, and with such a large domain, it was really crippled by inaccuracies if I tried to use the numerical methods and Mathematica to solve that as a partial differential equation. So what I had to do was use finite differences to approximate the spatial derivatives in the advection and diffusion components, and I used centered finite differences for that. And the result was from one equation that was continuous in both space and time. I derived a system of equations describing the transport of these velgers into and out of each of these individual discrete nodes over time, over the settling period, which is on the span of days. And in that way, I was able to simply integrate each of these equations over that settling period to find the the density of Velgers that remained at the very end of that settling period to find out how many would actually settle at each location to become adult mussels in that year. And this is the result this is in 1 year over the settling period here, which I had 7 days to look at kind of a worst case scenario. With the shortest settling period, you're going to observe the least amount of Velger mortality, just to kind of project the worst case scenario of the invasion here. So as you can see from this very high concentration that has been dumped into my system, you're spreading over space over time. And you get screenshots in time over there on the right. And this is when I integrated the two parts of the model, the adult mussel population growth model and the Velger transport model. So this projects how this invasion is going to proceed over 30 years. So you see, there are really only adult mussels in the first 4 years, and you have very high concentration here, up to 2,000 mussels per square meter in the first about 3 years or so. This is because once these Velgers are introduced at this first location during the first year, they settle around here, further downstream, and are about more or less normally distributed about the point, a point somewhere around here, due to the diffusion. And then from there, all the Velgers that they send out are swept right into the Chesapeake to die because of the natural barrier of salinity. So there are no Velgers settling back into the system, and that's why you get this